Welcome friends, welcome to Datta Biology class. Today our discussion will be on transcription. Earlier we have completed replication. We have completed DNA replication earlier. And after replication, we are going to transcription. It is a little bit complicated to that one. And what is the difference between this replication and transcription? Will come one by one. Now you see, in DNA replication, what we have seen, suppose it is a DNA. A DNA have two strands. Out of the two strands, another two strand will come. How? This strand will be their parent strand and another new strand will come. This is the daughter strand or new strand. From the other one, say this one we have completed. Now from this one will also be here like this, and new strand will come again recoil like this. So from this one single strand we have completed two strands. This was replication. What is the difference between transcription? Now you see, in DNA replication, the entire DNA splits up and 1, 2, 3, 4. Out of the new strand, these are the new strand. New strand has been formed and one DNA produces two DNA. It is semi-conservative and others that we have gone through. It occurs within the for total DNA, the total DNA replicates. But in transcription, the difference, main difference is that in transcription, suppose from this area to this area, this will be transcribed. Again, from this area to this area, this will be transcribed. This area will be transcribed. So in short pieces, it will be transcribed. Now, whenever it will be transcribed into a single strand. Here, the DNA replication we have completed the total DNA. In single strand, say for this one, this will be produced. For this one, this will be produced. For this one, this will be produced. In short segment, it will be produced. In short segment, whenever there is single strand, we call it RNA. These are called RNA. Because RNA have single strand. Moreover, here, the nucleotides are arranged in sequence. These nucleotides are there. So these nucleotides will also be there. These nucleotides will also be there. And it will be A, T, G, C, say for example, A, T, G, C. Here also A, T, G, C will come. But here, T will be replaced by U. Thymine will be replaced by U, A, T, G, C, U. So, from DNA to we have RNA producing, RNA has T has been replaced by U. So this is the difference, number one. Number two, one more difference is there. That is, in DNA, here the sugar was deoxyribose sugar. Here it is ribose sugar. What is deoxyribose and ribose sugar? We have come across again. I am repeating the same. It is C1, C2, C3, C4. This is O, and here C5. I have come across in DNA replication, just repetition. Here in the number 2 carbon atom, there was OH. OH group, whenever it has OH, it is known as ribose sugar. And if this is not there, it will be deoxyribose. So, from deoxyribose, this is produced, RNA is produced. This RNA replaces the thymine with uracil. And the sugar is transformed from deoxyribose to ribose sugar. This is the difference between replication and transcription. Transcription occurs in little parts, these parts, and it is the DNA which directs. I will come later. Actually, this transcription is synonymously called protein synthesis. This is known as protein synthesis. Forget about transcription. We are ready to produce protein. How? Now we will come into the transcription part. But before going into the transcription part, we have come across that it is known as protein synthesis. Proteins are made up of amino acids. Proteins are made up of amino acids. Now we should know what are amino acids. These proteins are linear molecules of several amino acids. Suppose this is an amino acid, this is an amino acid, this is an amino acid. These are all amino acids. These together forms a protein. These amino acids are actually the base of protein. How many amino acids are there? There are 20 amino acids. First you write it down that there are 20 amino acids, then we come time to time, really. Amino acids, there are 20 amino acids. Leucine, 1, 2, 3, 4, 5, 6, 7, 8. These 8 are called essential amino acids. These are the amino acids because today there is a trend of taking protein. So what protein does? We are taking protein, but protein is broken down to this. In these broken down proteins forms the pure protein, whatever is required in our body will come. Then these 8, there are 2 more histidine and arginine. Histidine and arginine, these 2 I have written in separate color.
Sir, can you tell all the names?
Yes, I am giving you. So there are 8. 1, 2, 3, 4, 5, 6, 7, 8. These are essential amino acids. These are semi-essential amino acids, and which are less required. Alanine, asparagine, aspartic acid, glycine, glutamine, glutamic acid, tyrosine. Last one is tyrosine. These are less required. These are less required amino acids, but these are required, less required. These are semi-essential amino acids, write it down. And these are essential amino acids. There are 20 amino acids. Out of this 20 amino acids, we will be producing protein. That is, in case of transcription, we will see, in transcription we will see that out of this 20 amino acids, we are producing different type of proteins. We require proteins what for digestion we require enzymes. The enzymes are made up of protein. Some antigen infects our body, we are producing antibody made up of protein. And protein for energy and others. So we are accustomed to, we are very much interested to take proteins. Now how these proteins are making from these amino acids, let's suppose for the purpose of our study, suppose Maggi is a protein. Say for, let us suppose, Maggi is not protein, but we have to produce Maggi. The protein we have to produce, which is required for our some purposes. Some hormones are produced, everything is produced, but it is for simplification I am writing. Maggi aim for, say, methionine. Methionine number, say for example, it is 5. This A for, where is A? Arginine 10. Say 10. G for, say glycine 15. G for 16. I for isoleucine, say number 2. So these amino acids are arranged in a sequence, in a specific sequence, in a specific order, so as to enable us to produce this protein named, we are calling it as MEG. Now, if from 5, 10, 15 and 16 are glycine and glutamic acid, okay? If you write, if the it is produced, it is 16 and it is 15, it will be different one. So it will be in a definite sequence. In a definite sequence, it will be produced. We come one by one. So proteins are made up of amino acids, 600, 700, so many amino, 600, 700 amino acids are arranged in a sequence of orderly sequence to produce that protein. Every time we are producing, we are taking protein and that protein is converted to, as per the requirement, producing hormone, thyroxine, producing enzymes, producing this. So these proteins are being produced day by day. In every moment, we are producing this amino acids. And these amino acids are arranged in a linear order. So proteins are known as linear polymer. Proteins are linear polymer of amino acids arranged in a definite sequence. Now, proteins are organic acids. Proteins are organic acid. This is the formula of acetic acid. Let me write. Acetic is CET acid. Acetic acid. This is the formula of acetic acid, not amino acids. In amino acid, H, C, this is glycine. In amino acid, the hydrogen atom is replaced by amino group. This is known as amino group. And this is known as carboxyl group. Clear? So amino acid is basic in nature and carboxyl group is acidic in nature. So in this amino acids, one of the hydrogen atom is replaced by NH2 by amino group. There are 20 amino acids like this, 20 types of amino acids. And it is the formula, in general formula is this amino group, this group and the side chain R will be there. There is a side chain called R. R is a side chain of one carbon atom attached to carbon atom, and there is NH2 group, and there is COOH group. This is left blank. So amino acids can be represented. General formula is R, CH, NH2, COOH. This is the general formula of amino acid, in which R is attached to a carbon atom. This R may be different. In glycine, this R is H. Write it down. In glycine, R is H. Clear? In alanine, R is CH3. In alanine, this R is CH3. Clear? And in arginine, R is CH2. In arginine, this R is CH2. So it varies. This R varies and the amino acid varies. So this is amino acids. Now we can say that one amino acid joins with another amino acid, joins with another acids, they joins. And this can be called, this is amino basic group, this is acidic group. So amino acid can be called amphoteric. What is known as amphoteric? AMPHOTERIC. Amino acids are amphoteric in nature because of the presence of basic group and acidic group. So alkaline and acidic group, both groups are present, so they are called amphoteric. The name comes from amphibia. So they are amphoteric. And now another two question. If this amino acid is having one more amino group, these are called amino group, amino acidic acid, amino acidic acid, or called basic acidic acid. These are called histone. Write it down, very, very important. Sometimes it comes. What is histone protein? That is, it is having one additional amino group, so it is known as basic amino acid, and the protein, subsequently the proteins are called histone protein. Likewise, if it is replaced by one more COOH, one additional carboxyl group is there, so it becomes acidic. So basic amino acid, those are called acidic amino acid or non-histone. Sometimes question may come, what is histone protein, what is non-histone protein? Histone proteins have one additional amino group or basic group, and non-histone proteins are having more than one COOH group. Now, we know that proteins are made up of various amino acids in a definite sequence. Now, how they join one with the other? These are two amino acids. How they join with one another? AH C NH2 and COH, NH2COH, CONH bond, CONH. Here CO will come and here from NH will come. What will be left? From NH, one H is left and from COOH is left. These two. So one amino acid remains joined with another amino acid by NHCO bond or CONH bond. This bond is known as peptide bond. This is known as peptide bond. Some enzymes are required, peptidyl synthesis will come later on, and for this bonding one molecule of H2O is removed. One molecule of H2O is removed and the two amino acids joins with one another with the help of peptide bond, and it requires energy. It requires energy. The energy is supplied by GTP and ATP. These two are required for joining of the two amino acids. So in this way, one amino acid joins with peptide bond here, peptide bond here, peptide bond here. That is, one NH2 group and COOH group joins one by one, because whenever we will see the transcription, then you will find it very difficult that how it joins. So this NH2 group of one amino acid joins with COOH group of the another amino acids which are binding by tRNA will come. So first we should learn that what is peptide bond. NHCO, this is the peptide bond, that is one with another, in this way they gets bonded by peptide bond, and in this way amino acids are linearly arranged in a protein, and in protein there are not 2 or 3 amino acids, 200, 400, 600 amino acids are there in a sequence. So it is very complicated and very minutely engineered. That is why we are doing all these things. So for transcription we have started amino acids, and next day we will start RNA. Next day we will start RNA. There are 3 types of RNA. We will go into the detail of all these things. One is mRNA will come. One is tRNA, and another is messenger RNA, transfer RNA, and ribosomal rRNA. These 3 are produced. These RNA, all these RNAs are produced from DNA. Will come slowly. So we go through what is our mRNA, how it is arranged, how it is produced, tRNA, how it is produced, what is the structure, and rRNA. After the completion of RNA, we will go to transcription later on. RNA and protein. This RNA, mRNA, they are actually produces protein, amino acids will come. So today we are concluding this, that we have completed amino acids. Next day we will start with RNA. Till then, take rest. Thank you.